Hello everyone, hope you all are doing well. So today we are going to do topic 66E, where we are going to discuss our topic 66E of paper 2, where we are going to discuss apple scab disease. Apple scab disease. First of all, although we are discussing here major diseases and pest and their management, so the first disease we are going to discuss today is apple scab disease. So its pathogen is Venturinia inequalis. Venturinia inequalis. Its symptoms are scattered circular brown spots with dendritic margin on under surface of leaves. Or you will also see dark brown spots in fruits. Dark brown spots on fruits. When you look at its occurrence, it is mainly found in USA. You will find this disease commonly in USA. In India, it is mainly found in Himachal Pradesh. Himachal in India. Now we'll look into epidemiology. In apple scab, the amount of initial inoculum is usually large, and released over, and this inoculum is nothing but ascospores. It is usually large and it is released over a period of 1 to 2 months following bud break. So after bud break, it is released for a period of 1 to 2 months in apple scab. So now, what are its causes? We all know that pathogen is Venturinia inequalis, but its causes, or you can say etiology. In etiology, whenever you are asked about etiology, you will just write the name of the pathogen, that it is caused by this pathogen, and how it is spread. You can write how it is spread. It spreads through on fallen disease leaves. In spring, the fungi shoot spores into the air. In spring, so this is a fungi. So this fungi in spring, this fungi shoot spores in air. And spores are carried by wind to nail, and these spores are carried through wind to places such as newly formed leaves, flowers, fruits, green twigs, etc. So this was its etiology. Now moving on to its management. How will you manage this? Management. So first of all, we will protect new trees or disease-free trees from infection. First of all, protect new trees and disease-free trees from infection. Secondly, keep plants healthy. We need to keep plants healthy. That is, we will provide water to them during dry periods. We will clean and destroy infected leaves. We will clean up, we will destroy infected leaves, and we will keep the area clean. Where we have cut plants, we will keep that area clean. We need to prune trees to allow good air circulation. Pruning for good air circulation. What happens? Good air circulation prevents infection. Prevents infection. We can also provide fungicidal sprays. Such as we will use sulfur, and you will use before spore has fermented, before spore germinate. Before spore germinates, you will use sulfur before spore germinates. We will use chlorothalonide, that is daconil. We will use mancozeb. We will use manab. So these are some fungicidal spray. We can also use, last point, resistant varieties. Such as some common resistant varieties are Florina, Freedom, etc. Freedom. These are the resistant varieties of apple, which prevent the tree from apple scab disease. So this was all about apple scab disease. Now moving on to the next disease, that is late blight of potato. Late blight of potato. Here the pathogen is Phyllophthora infestans. Now look into its symptoms. Its symptoms are bright brown and irregular patches. It starts from leaf tip. Bright brown and irregular patches. This starts from leaf tip and leaf margin. Later on turned into brownish black patches, and this later on turns into brownish black patches. Ground leaves show symptoms first. The leaves that are closer to ground show first symptoms. They will show the symptom as initial stages. In favorable condition, entire vegetative parts are killed within a day, hence called blight. Entire vegetative parts are killed, and when the conditions are favorable for the pathogen, then entire vegetative parts are killed within a day, and hence called blight. In favorable environment, infected area becomes brittle and detached. What happens in favorable environment? Infected area becomes brittle and gets detached from the plant. And in endogangetic plants, only mode of survival is infected tubers stored at low temperature. Only mode of survival is that infected tubers need to be stored at low temperature. So this is a very good method. Now, what are its risk factors? At which conditions enhances the late blight of potato? Risk factors of late blight of potato. These are temperature ranging between 15 to 20 degrees Celsius. High level of humidity. Humidity is high, and there will be excessively dense foliage. So in this condition, the risk factor increases. Water management, or you can say control measures. These factors enhance late blight of potato. Water risk management. First of all, we will use healthy seed material. Use healthy seed material. High ridging to reduce infection. We will do high ridging to reduce infection. We will do delayed harvesting because of high temperature. When the temperature will be high, we will delay harvesting so that it grows into cool condition. As we know, cooler climates is good for that pathogen. And what happens due to this high temperature? The above parts is dried away. The parts get dried. More what we can do? We will do labor intensive. Body X mixture is not used. As use of Body X mixture, or you can say Bletox 40, Bletox 50. The spray of these becomes labor intensive. This requires high labor. So these things are not used here. They are not used. What we can use is, let me rub it. In place of them, what we can use is, we can spray Mancozeb, or you can say Janeb at the rate 2 to 2.5 kg in 10 liter of water, and we will do 2 to 3 sprays at interval of 15 days. We can also do tuber treatment using mercury chloride, that is 1:1000 in water, that is 1 milliliter in 1 liter in water. We can also use resistant varieties such as Kufri Alankar, Kufri Jyoti, Kufri Chandramukhi, and this Kufri Chandramukhi is developed from CPRI Simila. So these are some of the resistant varieties. So this was the management of late blight of potato. Now moving to next disease, that is bunchy top of banana. Bunchy top of banana. Here the virus is banana bunchy top virus. And its transmission is, it is transmitted by infected suckers and banana. It is transmitted by infected suckers and banana aphid. What are its symptoms? Initially, dark green streaks appears in the veins of lower portion of leaf, midrib and leaf stem. Initially, what happens? Dark green streaks appears in lower portion of leaf. lower portion of leaf midrib and leaf stem. What happens? They appear to be bunched at the top of plant. They appear to be bunched at top of plant, the symptom for which disease is named, and as they are appeared for the bunched of the plant, that is from which symptom we have named the disease, the bunchy top of banana. Severely infected banana plants which will not fruit. The plants which are severely infected will not fruit. And but if sometimes the fruit is produced, the banana hands and fingers are likely to be distorted and twisted. And if fruits are produced, all of this are more likely to be distorted, or the fruits will be twisted. So these are the symptoms. Now moving on to the management of bunchy top of banana. First of all, we will use virus-free planting material. Remove and rugging of infected banana plants. Maintain clean, weed-free field for early detection of infection. Maintain clean, weed-free field for early detection of infested sucker. For the plant will be injected with 4 ml. We can inject the plant with 4 ml Pernoxone solution for vector control infection of plants. With for vector control, we will use monocrotopos 4 ml at 45 days interval. From 3 month 45 days interval, from 3 month till flowering. We will spray plants with phosphomidone. Phosphomidone 1 ml per liter. Or you can also use methylmidone, or we can also use monocarpotos. Here we can also use monocarpotos. These 2 ml per liter and monocarpotos 1 ml per liter. So these will also help. So this was management of bunchy top of banana. So that is all for today's session. For any queries, do comment. If you like it, please press the like button. Do share and subscribe. Keep making notes. That's all for today. Have a nice day. Thank you.